So it's a pleasure for us from the DOHAD Regional Society, Latin American DOHAD Regional Society, to receive today Dr. Yoko Nomura. She's a professor in the Department of Psychology at Queens College in the City University of New York, CUNY, a faculty research fellow at the same place, Advanced Science Research Center, and professorial lecturer in the Department of Psychiatry at the Mount Sinai School of Medicine. She's the principal investigator of a population-based longitudinal.
Oh, I don't hear you. Christine, we can't hear you.
Sorry, I think it was deactivated. I will start from the top. So it's a pleasure for us here from the Latin American DOHAD Regional Society today receive Dr. Yoko Nomura. She is professor in the Department of Psychology at Queens College in the City University of New York, CUNY, a faculty research fellow at the same university, Advanced Science Research Center, and professorial lecturer in the Department of Psychiatry at the Mount Sinai School of Medicine. She is the principal investigator of a population-based longitudinal research study in the developmental psychopathology known as the Stress in Pregnancy, SIP study. Funded by multiple NIH institutes, the SIP study focused on the gestational period during pregnancy, which is a critical but often overlooked stage for optimal child development. The SIP study has partnered with the New York Hospital Queens and Mount Sinai Hospital and recruited over 800 pregnant women. The cohort has been followed throughout their pregnancy and as their young child grew. This has enabled the study to uncover the interplay between child genetic susceptibility and psychosocial stress that mothers experience during pregnancy, taking advantage of the quasi-experiment of a human population. The study has focused on a variety of outcomes in infancy and early childhood, including early signs of psychopathology, suboptimal neurobehavioral development, HPA axis functioning, and gene expression in the past. Currently, the study is assessing the cohort who go through their puberty with molecular and neuroimaging focus. The overarching goals of the study are to help understand the impact of disaster-induced stress in pregnancy, chart the course of the mental and metabolic diseases from the in utero through birth and your early adolescence, and further understand the critical periods of development during pregnancy and early childhood. In addition to stress, currently the study has incorporated a geographical information system to address the impact of exposure to heat and air pollution during pregnancy on health development among offspring, to identify potential markers for suboptimal development during pregnancy and early childhood that can help determine whether interventions could preempt to developmental disease or impairment that might otherwise emerge later in life. So, Dr. Nomura, thank you very much for your presence here today. It's a pleasure for us to hear from you.
Thank you. Can you hear me? Yes. Okay, so thank you very much for having me for a DOHAD seminar. I run stress in pregnancy study. You said SIP study, what we call it SIP study. SIP study is a community-based birth cohort study taken place in New York City. As you can see on the screen, there was an orange color. It was exactly what happened last summer in August, in July, late July. It was due to a Canadian wildfire. And this is the first time public became seriously aware of the impact of the climate change and the global warming. Oh, I don't know why it's not moving. Sorry. I thought I was on a slideshow mode, but it's not letting me. Sorry about that.
Yeah, maybe you have to change the configuration if you can go out of the screen mode. Let's see.
Sorry. Yeah, it doesn't.
Yeah, I think you can click in the Ask button to finish the presentation, and then we change a configuration for the presentation.
Sorry about that.
When you see presentation mode, you can configure the presentation slides.
I think they're seeing it.
It's not.
Does it go?
Yeah, no.
No, it doesn't.
In the first one. And it's only a click in the configuration mode. You can keep on the screen, but just give an ask. Then I can see the configuration of the presentation. Yeah. So let me see. I think that slideshow probably.
Oh, yeah.
In the top of the PowerPoint, there's a slideshow. Yeah. Set up slideshow.
You set a slideshow before, and it's not
set up. Yeah. Then change for browse button in individual window. Let's see if it works.
Okay.
Maybe it's this.
Oh, now it works.
Now it's
Sorry
working.
about that, people.
Good.
Thank you.
No problem.
Then so that was the first. That was the first time. What is that? I hear somebody talking. So this is the map, the picture that prepared by National Oceanic and Atmospheric Association, which shows how many disasters had happened. In the last year, there are 28 natural disaster, weather-related disaster, each of which cost more than $1 billion. As you can see, various agencies have recorded that land temperature is on the rise. NOAA, as I told you, National Atmospheric, Oceanic and Atmospheric Agency show land temperature is going up by various indices. And also, this is the sea surface temperature. Sea surface temperature is also on the rise. And sea surface temperature actually is, oops, what happened? Actually is the one which is an indicator for the magnitude and frequency of the natural disaster, such as hurricane and hurricanes. It's not going anymore. Okay. So impact of climate change, we know there's a huge impact on health, mental health, physical health, as well as community health. If you can think about it, if $1 billion, total of over $100 billion have been spent in a year, that money could have been spent for welfare or enrichment for people's life. But that money every year has to be spent for infrastructure recovery. And if you think about it, that of course is going to decrease the quality of community health. There are also certain vulnerable population, people with disability, elderly individual, children, and also fetus. Oops, how come it's not changing? Fetus, fetus meaning pregnant women are also part of it. Those people are vulnerable because those people at the impact of climate, impact of stress, impact of environmental sources have a profound influence on their trajectory of development. as you know, that people, your audience knows DOHAT theory. The people who don't know DOHAT postulate the environment stress in utero early development influences health and risk of developing non-communicative disease, such as diabetes and the heart disease. But over the time, we realize it's not only a non-communicative disease. Psychiatric and neurodevelopmental disorder is impacted. Early studies show that the focus of DOHAT theory initially was malnourishment, not having enough food, and stress. But over time, we realize it's have a lot of anything which has associated with a poor in utero condition have a long-lasting implication for people's health, especially with the brain. Early study used low birth weight and the preterm birth and those kind of easy target as a proxy for poor in utero condition because we didn't really know how to measure it before. And we didn't really know what happened in utero. Oops, it's not moving. Okay, so this is a power up version, more updated version of the DOHAT theory. Primer, the old proxy, low birth weight, preterm birth, low Apgar score, and the small head circumferences, which are often used as a proxy for poor in utero environment, have been replaced by potential underlying mechanism. Alteration in the maternal fetus endocrine milieu, IUGR, it's the intrauterine growth restriction, small for gestational age. And the epigenetic regulation, the oxidative stress, and the inflammation are one of those focus. And thanks to over the past 3 decades, thanks to the scientists in different fields, transcending disciplinary boundaries, we are working together to really tackle what's happening in this in utero period. So over the decade, we also realized not that we used to call it poor in utero environment, but we know there are 5 major categories: maternal nutrition, maternal disease, environmental exposure, stress, and the maternal lifestyle factors, such as using substance or not exercising enough on that kind of thing. In our study, we have looked at food restriction. We have looked at infection, gestational diabetes, and the preeclampsia. Gestational diabetes and preeclampsia are known factors which pregnant women, pregnant individuals often have, but have a profound implication for brain development of the offspring. The heat and poor air quality is something we are looking at right now. And the stress, we use the Superstorm Sandy as a natural experiment. As you can see, in human population, we cannot really manipulate the condition. We cannot randomize people to different conditions. That is called randomization, which is a gold standard in research. But we can, as you can think about, when baby is born, we cannot swap the baby to a good mother and bad mother. We cannot put the mother in a tank, water tank, and make them swim until they give up swimming and that kind of thing. In human, it's not possible. The second best thing we can do is a quasi experiment, taking advantage of natural disaster. So you will be able to create a quasi experiment of the women, of the individual exposed to certain conditions as opposed to the other. And over the decade, as you can see, they are looking at the different organ system because different organs have a different different factor have a different implication for different organs. in our study, we focus on brain, we focus on the neurodevelopment, but cardiovascular, reproductive system, metabolic system, everything else might have a different implication, which we don't study, but I wanted to point it out. So this is how it goes. When I was collecting, I have a career development award from the National NIMH, National Institute of Mental Health, which really trained me to be able to conduct epigenetically informative community-based study. And at that time, epigenetic was in its infancy, and incorporating epigenetic information in a community research instead of the bench research was a big, big issue. And I was trained in that. And as I was being trained, Superstorm Sandy happened. So I have an infrastructure already set up. I have a training. I understand the neurodevelopment. I was at the right place at the right time with the right skill set. So we have formulated this environmental, the SIP study, which allow us to collect placenta tissue, cord blood, and cord blood and meconium, so that we'll be able to really understand the connection between what happened in utero and how gene expression changes. As you all know, DNA doesn't change quickly, but epigenetic does. Epigenetic is a really important areas of research, because if we are not able to target the modification or the changes, there's no point of us studying, because after all, we are serving for human people in the community. We want to be able to make their life much easier and have a better trajectory of development. So this is how I formulated the study. In the middle is an exposure to Superstorm Sandy. So women are pregnant during Superstorm Sandy. And people sometimes ask, why Superstorm Sandy? It's really is a too simplistic categorization. It's not. Because what I am trying to capture is a connection between mother and the fetus. And the only placenta and the cord is umbilical cord is connecting the two individuals. So even if we say Superstorm Sandy, yes and no, it's really means control groups are not tied to mother with umbilical cord. The control people are individual outside of mother's tummy when Superstorm. So stress is, they are not sharing a stress, because what happens is whatever mother experience is going to pass through placenta, going through the cord blood, and given to the fetus. Over 10 decades, not 10 decades, 1 decade, 10 years, we have collected parallel measure of a lot of different things. Around the 5 and 8 years, we started implementing brain neuroimaging study. We have studied the pilot data, collecting pilot data in order to have a full-scale neuroimaging research. And currently, we are doing it. We have neuroimaging, and we have epigenetic, we have developmental indices. And another important thing is our study is epigenetics is, just think about the driving. When you are driving a car in a straight line, there are less likelihood for accident. But when you are turning intersections, that's when the accident could happen. You can think about epigenetic in that way too. When things are changing drastically, that's when the mistake could happen. So in utero period is one important time for epigenetic research, and puberty, when children are changing into men and women. Their shape is changing, their social role is changing, everything is changing, and that creates an unparalleled opportunity for us to understand that the epigenetic implication. Not only that, we want to be able to connect behaviors and brain. How behavior and brain are going have been governed by early prenatal epigenetic modification, and how postnatal environment continue to influence those behavior and brain indices. Okay, a pregnancy, we looked at the ambient temperature and the psychosocial stress, and among other, with everything else going on, and those are our current focus. At birth, we collected those biospecimens, and the research nurse who are on our team. We have a full operation at the Mount Sinai OBGYN clinic, and all the labor and delivery nurses are collecting data for us at that time. So we collected vital record, one of those the easy birth weight, gestational age, Apgar score, and all that. Starting at 6 months, we started collecting temperament of the children. Temperament is a really important indices for one of the important indices for children's neurodevelopmental profiles, because children's when they are younger, we really cannot really tell if they have this disorder or that disorder. Do they have a problem? Is, for example, if you are going to identify ADHD or dyslexia, learning disability, you cannot really tell if that person is having a problem when you are dealing with one-digit multiplication, one-digit summation, or division. It's only become more apparent when you are dealing with two-digit, because you have to have it in your working memory and have to store the information. And then that's when you are going to be able to really kind of detect something is happening. But we don't want to wait until late, because early detection is everything. Early detection really prevent people from additional suffering, which we could avoid. So temperament really is a really good index to really find out what might be happening in infant. Starting at 1 year and a half, we started collecting information about clinical problem and competency or adaptive behaviors. And we started having a structured interview to come up with a diagnostic outcome, psychiatric disorders. And there is a myth among all of us that children don't have psychiatric disorders. That's a myth. Children do have psychiatric disorders. We just cannot really see what exactly what's happening, because children are not able to, their brain is not developed enough to be able to articulate what they are experiencing and what they are really feeling. So it's difficult, but it's possible to just with a targeted structured interview, especially for prenatal children, not prenatal, preschool age children, we are able to have some diagnostic outcome. Starting at the same time, we continue to collect biospecimen, and we have the psychophysiological experiment. And this is just an example. The panda bear, mom and the baby bear are playing nicely, and out of the blue, mama just sneeze with a big bang. And that's supposed to create a startle in children and mother. And that's what we recorded. As you can see on the diagram below, we have 5 blast, and each time we measure the elevation of heart rate and electrodermal activity, which is a sweat on your palm, and respiratory sinus arrhythmia, which is when you get anxious, you breathe like that, and that was captured. Starting at this age, we have started collecting neuroimaging. And as you can see, this is how we do, and we have an eye tracking paradigm. We track the eye movement. So what are we doing now? use placenta functioning to link behavior to the brain. As I told you, placenta is very unique organ, and it have a lot of information regarding placenta, regarding epigenetic functionality. So that can really tell the differences, that how it influences the behavior and the brain, and how if brain come first, chicken and the egg kind of, brain come first or behavior come first, or both of them simultaneously influenced. And we are able to do a lot of different things. And the underlying persistence influence of the supersonic exposure in utero, which we see over and over again. We saw supersonic exposure would end around the time of infancy. It's not true. It has a profound influence throughout adolescence, even at this time. It is one of the biggest, strongest risk factor for suboptimal neurodevelopmental development. And we want to show connection between different pathways using molecular, psychophysiological, hormonal, behavioral, brain function, neural functioning to just come with one human being. That because human, you know the story about elephant, right? The elephant is in a room. It's dark, and every blind people touches different part of the elephant. They report, this elephant is a table, this elephant is a carpet, this elephant is a rubber band, or depending on the area the blind man touches, they come up with a different thing. And the same thing is true. We should be able to, we have to really look at the specifics of the development, but we also have to be able to put them together and understand the person as a whole. And last thing is testing double hit and stress inoculation model. Those are the two major stress paradigms. One is double hit acceleration, meaning if you hit once, the second hit is exponentially impact is much higher. So second time, you might die. The inoculation thing is if you are exposed to mild stressor, maybe manageable stressor earlier, you become stronger. There is a song that whatever doesn't kill you make you stronger or whatever, right? That's as that song tells. If you are exposed to something and learn how to deal with it, you become stronger when something else happens, and your body knows how to manage it. So those two stress paradigm, they shouldn't be mutually exclusive, but we want to be able to test it, and that's what we are doing. Okay, there are four major big project going on in our lab. One is examining the role of placenta. Those are the major players right now. Dysregulation of the gene associated with HPA axis and immune network in offspring is related to mother's prenatal stress or children's reactivity and stress responsibility, or executive functioning, lower executive control in children, or does socioeconomic status or something, opportunity and resources, does it influences the association between those many questions. Project number 2, we are looking at the heat during pregnancy on the health of mother and the offspring. The weather, the heat, especially heat and the pollution, but heat is really influencing the neurocognitive development of the children. That is a question. I used to say to people, I really didn't understand what is happening, but egg is being cooked, maybe. That's how I described. The mother's eggs are being cooked, and when you see the hard-boiled egg versus raw eggs are much more fluid, and the hard-boiled egg is more solid. And we want our brain to be fluid. We want our brain to be plastic, a brain to be able to adjust to whatever the input we are getting. And I wonder the heat is going to make that programming in whatever the molecular level we have in our body. That was how I started this project. And also another thing is there any vulnerable population? Because heat doesn't affect everybody equally. As you can see, the rich people can get out of the city and go to the second house, country house, and get food. Or the children might, the people might have different level of access to air conditioners. And so it's not really equal. It have a differential implication. Heat has a differential implication. How about additional law of air pollution? Because heat doesn't happen alone. Heat creates a dome, heat dome, and within the heat dome, the pollution is going to be captured and trapped. So you are likely to be exposed to a higher level of pollutions. That is a problem too. So we have to look at heat and pollution at the same time. Project number 3 is looking at the brain functioning and the brain connectivity and brain's volume structure. Amygdala and frontal activity are different. Basal ganglia activity is different. And frontal connectivity between, functional connectivity between frontal limbic region. Limbic region is known as some of the people who knows about brain research. Limbic region is emotion core, right? The frontal region is an executive core. You make a judgment, that adult decision. Emotion core is a limbic area. And the project 4 is geographical information system. We use geographical information system to collect the data at the macro level. So pollution level, noise level, socioeconomic status, having a lot of supermarket, how many park do you have? That kind of thing, information can be obtained there. Okay. This is a picture of the fetus. So those are the 4 projects. And this is a fetus. I just want to go back to where I come from, epigenetic research using placenta. As I told you, placenta is a really unique tissue that connect the interface between mother, mother who had developed brain, and the fetus, which has a developing brain. And it's a unique tissue because it's discarded at the birth. It's not wanted tissue. It will be thrown away. It's go to the morgue. Nobody pay attention to the placenta, but it has really a really important implication. And I'm going to just show you this. Each cell has a set of the expressed and the gene have to be silenced or expressed. And during the fetal development, oligodendrocyte progenitor cell have to be converted into a mature cell in the developing brain of the fetus. One of the well-known epigenetic mechanisms, histone modification, which shows there, is important in this period. The long DNA can fit in a tiny space going around the histones, and you need epigenetic programming for oligodendrocyte progenitor cells to differentiate and marinate in the central nervous system. They are so-called, those are TAG, which is coming as the environmental exposure. They are so-called lighter than erasers. Lighter express the gene, and eraser silence the gene. And the balance of lighters and eraser define the epigenetic landscape of the central nervous system development. And as we know, brain doesn't happen at birth. Brain is being in production for a long time during pregnancy. So it means what mother had experienced and what mother had felt during pregnancy have a profound implication for the direction of the brain development in the fetus. So this, I just wanted to show you, this is based on the placenta. We have done RNA-seq. RNA-seq really captures all the RNA expressions and the cluster the genes. So I'm going to show you one at a time. This shows syncytia trophoblast, STB. That's the most important cell, I would say. That's the outer layer which separate mother and the child, the mother and the fetus. As you know, fetus really is an alien to the mother. During pregnancy, mother's immune functioning is going down to really, really low, just because we have a mechanism which allow mothers not to kill the fetus, because it's an alien. If it's an alien, an invader, mother's body is going to try to kill the baby. So in syncytia trophoblast layer is really a protector between mother and the child, and it's associated with the exchange in nutrient, gas, waste product, and the drug or stress hormones, because it goes through the placenta. And when we look at the placenta tissue, there are many, many different cells, but syncytia trophoblast cell is the only one which differentiated the supersome Sandy status. So people who are exposed to supersome Sandy have a lower syncytia trophoblast percentage, which means, remember, syncytia trophoblast is the one which is separating those two, the more the better. So it shows that, you know, it's not good. And this is the heat map to show the differentiation. It's really, really highly clear. Blue is the placenta of the mother who are not exposed to the superstorm Sandy, and red is mother who are exposed to superstorm Sandy. The clear division is really utterly astonishing. And enrichment analysis identifies 3 genes. NR3C1, people who are have molecular knowledge know that this gene. This is a usual suspect. This is a glucocorticoid receptor gene. Glucocorticoid is the one which helps secrete cortisol. So it's a stress hormone. So though it really makes sense. And the RUNX1 is a pro-neurogenesis gene, and the last one is intolerance and the immunity at the maternal fetus interface. So that is, we are very pleased to find those 3 genes to be enriched. But since we are studying the stress, we looked at the differences between those 2, as you can see. The superstorm Sandy exposed people have a higher expression of stress hormone genes. And when we looked at the association between genes and the syncytia trophoblast percentage, there is a negative correlation, meaning if you are less stressed, you have more syncytia trophoblast cells. So this initial the RNA-seq analysis really identified there is something which is important, biologically important, molecular biologically important consequences of exposure in utero. Now moving on to the brain, and overall goal is to see the differences between stress exposed and the heat exposed children in utero, fetus in utero, who have looked at the brain functioning. This is amygdala responses to negative facial expression. This is a typical, paradigm. You show many faces, and some faces looks angry, some faces look happy, some faces are neutral. but people really identify. So you probably know some of your friends are really like a very sensitive to anger or negative feeling of others. So they see neutral face as more negative. Oh, they look angry. They look sad. They are unable to identify neutral face as neutral. They are likely to see it as more negative. And that really is related to amygdala activity. Amygdala is an emotional brain. And as you can see, the children's brain who are exposed to supersonic sounding in utero has a higher amygdala activity, both right and left. When we looked at rostral and anterior cingulate among those exposure, same thing happened. And this area is, I did it again. Whenever I have a ruler here, which is preventing me from doing things, how do I go back to where I was? Oh, good. The affective response, it's controlled affective responses to conflict and error. So same thing. They are sensitive to conflict, and they are sensitive to making error. The processing of conflict and processing of errors. So we are pleased with that too. Oops. It's not moving anymore. Oh. I don't know why it's not moving. It's not moving. It doesn't go to the next slide, and Shamila is not here, right? It doesn't go. It doesn't move.
Can you put escape, please?
Escape?
Escape, I think that it would help to run, no? Or no?
Maybe in the bottom has a place to
Shamila
put
came
in
to
the next slide. In the bottom of, yeah. See if it works.
Oh, thank you so
it's
much.
going.
Okay, so this is a basal ganglia brain volume. Basal ganglia is known as a motor functioning, but nowadays we know it's also control processes such as emotional, motivation, and habit. So it also makes sense for the green bar is a supersonic exposed children, and the blue are normal, the not exposed children. And it shows the same pattern. So we are really, the brain side of the research is still in our infancy. We are still collecting data, so those are preliminary, but it's go in a direction where we know we want to see. And now moving on to the third window of susceptibility research. Over here, we are looking at the heat exposure at the first, second, and third trimester, and see if there is a magnitude of risk elevated among those who have less resources. As we wanted, anticipated, we didn't know that risk in a specific trimester, but there is an increased risk of gestational diabetes mellitus, hypertension, preeclampsia among those who are exposed to supersonic sounding in utero during first trimester. Everything is first trimester. And as I told you before, gestational diabetes, hypertension, preeclampsia is in mother, which happened a lot during pregnancy. That is a really big risk for suboptimal neurocognitive development of the children. We look at the birth indices, birth weight and gestational age during the first trimester exposure is reduced drastically. And then we look at the clinical problem and competency at the age 2. Same thing happened in the first trimester exposure. How about clinical problems significantly greater than other groups, but also competency went up. This shows the double hit is not part of it, but it's really indicate the fact that we really should look at clinical problem as well as adaptive behavior simultaneously, because it's clearly show while clinical behavior is elevated with a risk factor, adaptive behavior also get elevated. And what happened? We look at the stress hormone, DHEA and melatonin. Unlike other findings, DHEA was associated with elevation in the second and the third trimester. However, melatonin is associated with all the trimester, the highest in first trimester. We don't know what it means yet. We are going to really try to understand what is the underlying mechanism to see those findings. And as I told you, especially we are living in New York City. New York City have this bubble. City have concrete. City have the tall building, and we are dealing with heat much more because everybody uses the air conditioner, and everywhere becomes so hot. We have to use the air conditioner, but air conditioner produces heat. It's a heat. If you are walking on the street, air conditioner's exhaust is so hot. So as I told you, heat create the bubble. Heat create a heat dome. Underneath of the heat dome, air pollution get trapped. So this is the things I have looked at. Steroid hormone in hair among children at age 3. What I was trying to show is the top and bottom. Top is the main effect alone. Bottom is the interaction between the two. So heat TM3 is a trimester 3 heat exposure, and 2 is second trimester, 1 is first trimester. PM are particulate matter, small dust, and NO2 is a nitrogen dioxide, the air pollution we target. And interaction between those, among those things, shows that it's elevate the risk for elevation of the cortisol and progesterone. Progesterone is a female hormone, which the few weeks ago, there was a report by the BBC, which had been always talked about before, that air pollution is making the female children's menarche much earlier. The first age of menstruation is happening at an earlier time. And this may be this is going to give us a clue why it happened or how it happened. I think it's an important line of questions because that is also related to the neurodevelopment in the future. And since interaction is difficult to interpret, I just make it easy for you to see it. So we have high and low NO2 and PM 2.5. As you can see, in the high group, the risk, the heat and the risk elevates. So we can really see the differential association between high and low group. So this is how we interpret the interaction. And this is I just wanted to show it really is happening. And this is a gestational diabetes with particulate matter 2.5. And this called distributed log model. So we look at the whole changes across the pregnancy and see when is the highest risk for gestational diabetes. And as you can see, the line 1 is a neutral. anything which go above 1 is the risk. And you might think, oh, the risk in the index is so small, but it's not really small. But because we are looking at unit of analysis is small. We are looking at the particulate matter in a continuous measure. So it's elevated really, really highly. And that is a time during second trimester is a time for susceptibility. Okay, so I told you many, many things. And the reason why everything is so brief is because we have so many findings and we are really trying to gear into combining heat, air pollution, and the psychosocial stress in pregnancy and puberty we are dealing with. Is there an equal burden for families with marginalized group and with lower income? That is another thing we want to look at, because those different, the association could be really different among different groups, vulnerable population. So what do we do? Those our findings across different disciplines demonstrate that psychosocial stress, psychosocial factor are amplified by the effect of systematic inequity. And how can we start getting into this line of research and try to be an advocate for people who are really need assistance? So this is Child Opportunity Index, which Shamila, one of our doctoral students, have looked into. Child Opportunity Index, unlike SES, socioeconomic status, cover a lot of important area that it's important for the children: education, health, and social and economic status. And one of the things we are focusing on is this environmental pollution. And this is something which I put out to convince you. As you can see, its Child Opportunity Index distribution is really like, it's kind of scary to see. Asian and white and Asian have a higher Child Opportunity Index, whereas Hispanic, Black are really lower. This is the one which we really have to demonstrate, and I see this have an implication for children, our generation, the future generation, and also how we can just make it more equitable to people and get people understand we really have a lot to learn to really support those group of people. Okay, the summary. Stress during pregnancy have a negative implication in the brain area related to emotion regulation and connectivity. Heat exposure in the first trimester is the highest negative implication. Air pollution seems to be the second trimester. Remember in the diagram, I said, depending on the area we are looking at, or depending on the risk factor we look at, the susceptible window may differ. And this really shows we have a lot to learn, and we could really focus on trimester-specific exposure and see when is the most important time of the exposure. Next step, to understand the impact on epigenome, especially heat protein immune functioning, to estimate their impact on the brain development and from limbic connectivity, to identify vulnerable population, and to understand interplay among elevated risk, elevated heat, air pollution, and stressful condition. This is an old picture. I'm sorry, folks. This is a study team, and we are those are our collaborators, and those are funding, and this is our reminder. climate change is really happening. We really should find out the way to tackle that into our question, and we can do that together. Thank you for your attention. And sorry about many technical difficulties. I will take questions.
It was fantastic. It's so impressive to see these results. And when you can see like the modification in the amygdala, in the function, but also in the structure of a so important area of the brain, it's really impressive. We have questions from our guests. Let's see. So I have one. When you evaluated, oh, sorry. Please, you can open your phone and make the question. Yeah, later I'll talk.
Hey Yoko, this is such fantastic work, and I'm a big fan of your work, and really love that you're looking at multifactorial approaches. It's so important to show this transgenerational effect you see on children, on fetuses, that then leads on to their lives as it moves along. It's such important work, and thank you so much. This was such a great presentation. I have one question. This is something I hear a lot in this context, and it is this work is so important to be able to, like you said, like to be able to bring about change in the policies and so on. And sometimes the pushback I get about these great studies is that correlational aspect, and people and researchers or scientists looking into, but what is there causality? Like, how can you show the causal link of like what's really happening? How do you answer to that? Do you think it is necessary to show causation when we see so apparently the links between the environment and how it affects children's brain development?
I don't think there is a way to convince everybody to demonstrate the causality. So what I do is we have collaborators who dealing with animal model. So what we found will be given to the animal researcher, and they are going to test exactly what it is, and they are going to look at the brain tissue, and they are going to come. We go back and forth between animal model and the human model, because there is no way, even if we know for sure, this kind of obvious that there has to be a causality. And when we look at the timeline, we can comfortably say there is a causality. It's not the association, but I am really, really careful not to claim causality. In human research, there's no way to just 100% confident about causality. We can always have some experiment. We are in a lab, we are beginning to have this experiment in the stress paradigm, because we want to see the changes, but I don't think it's possible in human research. So we have to find a way to collaborate with animal researcher and go back and forth between the two. And I know what you're going to say. One drawback between translation research between animals and the human is a grant doesn't really support the translational research like that. They want animal only and human only. So it will be really difficult in terms of obtaining funding and do right things in a proper way. So what we do is really try to just patchwork together and try to do something in human, and then animal people get something, and then they get their own funding. We cannot really combine them. When time comes to combine animals and the human research, that would be fantastic, but I don't think it will happen in my lifetime. I don't know if it's a good answer, but that's how I think.
It's a perfect answer. Thank you.
So did you evaluate any interference of the diet for that experiment that you evaluated cortisol and progesterone elevation? Because they are steroids. So maybe they're coming from cholesterol and they can be impacted by the diet of the mothers or their own diet. I think that their own diet. Did you evaluate some of these aspects, the nutritional uptake?
I have looked at the intentional caloric restriction that, unlike Dutch famine era, nowadays young women, young individuals who are pregnant are under tremendous pressure to be thin. So they really don't want to gain so much weight. So they intentionally restrict calorie and try to stay slim even during pregnancy. And I have looked at the impact of that on the neurodevelopmental the indices, but we corrected the food intake, but it's based on the self-report, and we added it afterward. So we haven't even looked at the food intake, the influence of the food intake on the hormone level yet. But you are spot on. We have to be able to. We are thinking about doing the big panels of those cholesterols and the triglyceride and that kind of things, and I see how we are able to identify the implication, but we haven't looked at the food area yet. Somebody should do that.
Interesting, because the activation of the HPA axis could be related to this increment, and could be related by the diet. I don't know, maybe it's just.
I think so too. Diet have to be part of it, but as I confess, our diet data is not that great. As I told you, it's based on the self-report, and we don't really know if it's how accurate it is.
True.
Yeah.
Yeah, I understand. We have a question here from Sandra Sukasa. I wonder if there are any research about how does chronic stress influence on these aspects? So not like an acute stress, but chronic.
Chronic stress in pregnancy or chronic stress in a postpartum?
Let's see, Sandra.
In pregnancy. I think the stress have to be measured in very different way, like perceived stress or received stress, or the objective stress versus perceived stress are important matter, and chronic stress, chronic strain is another important stress, important areas of stress construct. I was trained in stress, psychosocial stress, when I was doing my PhD work. So I was trained to measure different aspects of stress one by one, and chronic stress is one of the major important stress. But I focus on this quasi experiment. So I have only been looking at Superstorm Sandy related stress, and we really should look at those chronic stress or ordinary stress. And we have just started looking at the interaction between Superstorm Sandy stress and ordinary everyday life kind of stress and hassle. But I just agree. I agree with you. It's important, but we haven't done it yet.
Okay. So anyone else? I have more questions. I keep talking here. So Dr. Yoko, did you find any gender-dependent effect, sex-dependent effect? Because I usually work with animal models and with rats, and we found a lot of sex-dependent effects, especially within exercise and diet used during pregnancy. And we have a total different effect on the offspring when we are studying male or female, including the cognition. Like exercise during pregnancy is much better for male offspring than female offspring. It's really interesting that we have this kind of differences in our models. I don't know if you have evaluated this kind of aspect.
sex difference, especially in this age range, is really, really important. And whenever possible, we look at the differences in the sex. And one of the person who is looking at the child opportunity index, Samira, I don't know if she's still there. Samira have looked at the sex difference, especially with interacting with air pollution. And I also have looked at, I have published a paper with a year ago, which generated so much controversy in the area that which found women, the female children, are more likely to have anxiety disorder when you are exposed to Superstorm Sandy. The elevated risk and the early onset of anxiety is so profound among female children, but for male children, it was attention deficit hyperactivity disorder and conduct disorder. So there is definitely differences in female and the male, but the reason I didn't present any sex differences today was because I am basing on finding in our new wave, and we only have 73 participant analyzed, and the number is small, so we won't be able to look at the male and female differences, but sex difference is so important. And I will look at it. And especially with the steroid hormone, we are changing, the children are changing. And so it's important to look at them separately.
Oh, yeah, for sure. Thank you, Dr. Yoko. You have any more questions? No? So, Dr. Yoko, would like to thank you so much for your time, for your knowledge, for sharing all of these impressive results with us. And thank you very much for being here today with us. We enjoy it a lot.
Thank you very much. I hope to see you in your summer.
Yes.
They are in winter now. Okay.
Yeah. Thank you very much for everyone to be here. Erika, I don't know if you would like to make a call for our course. We have a new course that is ending the registrations tomorrow. It's Life Cycle and Doha. It's free for postgraduate students, and it's only $20 for professionals. The inscriptions, the registration is open to tomorrow. So if you didn't get it, please get it now. It's being organized by me, by Erika Chavira Soares, that is here, Fabiola Suano, and Paola Casanello. I think all the 4 coordinators are here. It's a course for all the Latin American students, and we have almost 100 students enrolled in the 2nd edition.
That's great. So if you can send me the link, I will share it with our group.
Nice, great. The course is in Portuguese, Spanish, and English. We have 3 idioms, 3 languages. It's really interesting because a part of this, we understand each other. So it has been really nice to organize this course. It's our 2nd edition, and it starts now in August, in the 1st week of August. I will send you the information.
Okay, thank you so much. Okay.
So,
okay, so thank you so much for having me, and good luck with everything. See you.
Thank you. See you. Bye-bye.
Thank you so much. Have a great night.
How do I leave? How do I leave?